Okay, so previously we started with quick recap of your previous classes. So we started with the introduction to biomedical instrumentation. We started with the block diagram, operational modes, and then different physiological parameters, and then classification of medical instruments, and then we stopped with biostatistics. So regulations, I'll take it at the last. So now I don't want to take that separate track. Okay, so I'll discuss this in the upcoming classes. So next section is biopotential and biosensors. So when I say biopotential, we all know that our body is made of nervous system, right? So nervous system will transmit the electrical impulses from brain to all the parts and spinal cord to the all parts, right? So the brain and spinal cord are the main parts of your central nervous system. So all the organs and tissues are supplied with neurons. So these neurons will transmit some electrical impulses. That electrical impulses will cause some change in the chemical and the chemical action, and that chemical action will result in a biosignal. Okay, so that's what is an action potential. So let me just quickly show this video to make you understand the better. Are you able to hear the sound? No, right?
Hmm. No, ma'am.
Okay, just let me share it again. I think I should have this tab. Okay, yeah. So this video is a short video where you will be able to understand the action potential very clearly. I have all the pictures, but even if I explain it in pictures, this one single video will make you understand it in a much
Integrate
better
said
way.
information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. This is a neuron, which has four main parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmits the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, the dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass that signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon in a phenomenon called an action potential. When this happens, we say the neuron fires. Transmission of a neuronal signal is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed between the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, We also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called an electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, a neuronal membrane has a resting potential of approximately minus 70 millivolts. This means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the
Okay, let me just pause it right there. We'll get back to this video once we understood the concepts better. Okay, so let me share the PPT. Yeah. So this is the PPT for biopotential. So to get a quick recap of what we have discussed so far, we have previously seen the block diagram for the medical instrumentation, right? So you have a measuring sensor, signal processing unit, and all those stuff. So when we say computer aided diagnosis, we use a computer that will actually detect these biosignals and help the doctors to say whether the person is having a disease or not. So if you are going to look at that in a block diagram way, first you have a patient. So the patient has a physiological system. So he has numerous organs and tissues. So from the patient, you are occurring the biomedical signals, and the signals are occurred by the transducer. So what the transducer will do? It will convert one form of energy to another, right? So once the transducer receives the signal, it is sent to an amplifier circuit. So we have preamplifiers, amplifiers, and filters. So preamplifiers are added before the amplifier circuit. So this and all we will discuss in the second unit. So how the amplifier is designed, all those things we'll be discussing in the second unit. So this is a signal processing circuit. And then you have an analog to digital conversion. So all these signals are in analog form. You are going to convert it into a digital form. When I say analog form, in analog form you will just have the numerical value which cannot be electronically processed. So when you convert that analog signal to electronic signal that can be processed by a computer, then it is called a digital signal, right? So you are going to convert it using an ADC converter. Okay, so this conversion alone you will be studying in a separate course called analog and digital communication. So that's for that course. Okay, so we won't be discussing more about that one. And then you have some filters to remove the artifacts. So whenever you're dealing with signals, you will use a filter which can give us the exact data. So for example, if you take a tea filter or a decoction filter, whatever it is, so usually in a tea filter, what that tea filter will do, it will filter the tea particles and it will provide the tea at the bottom, right? So similarly in this circuit, a filter will give you the exact signal what you are looking for. For example, if you are going to take the ECG signal from the heart of the patient, okay, so if you are going to take the ECG signal, there won't be just ECG alone in this all these signals. Okay, so ECG will be mixed with other movement artifacts. If the person is breathing, then breathing also will disturb the signal. Okay, and if the person is running or if the person is doing any other activity, if the person is sleeping, all those things will cause some disturbance in your actual signal. So in that case, you need a filter, and then you use a detection algorithm. So when you give all these signals to a computer, you use some algorithm so to detect some events, whether the person is having any heart attack or if, whether he will be getting an heart attack, like that you will detect and predict some events, and then you extract the features, and then you do the pattern recognition, classification, and diagnostic decision. So these three blocks you will be studying in your signals and systems and biosignal processing course. Okay, so this is how a computer aided diagnosis is done. When I say diagnosis, you are going to find out whether the person is normal or whether the person is healthy or whether he is having any disease. So diagnosis is you are going to find out whether the person is healthy or not. So when I say computer aided, your computer is going to help in finding out whether the person is having disease or not. Okay, So the first four blocks together is the data acquisition and signal processing block, and these two comes under signal processing, and these two are the signal analysis block. And then all this data is given to a medical specialist or the doctor, and the doctor will give out his final prescription to the patient. Okay, so this is the computer aided diagnosis. So for all these things to happen, first step is you have to acquire the biomedical signal, right? So you have to take the signal from the patient. So to acquire the signal from the patient, you need some sensor and a transducer, right? So how the signal is produced, how it is generated, that and all you are going to see in this first section. So first of all, your body is made of in a electrolyte. So the cells inside the body have an electrolyte which is rich in potassium, magnesium, phosphate, sulfate, bicarbonate, and even small quantities of sodium, calcium, and chloride. So the human body cell will have all these chemical components. Okay, and the main function of this electrolyte is to provide inorganic chemicals for cellular reaction. So in anatomy and physiology, you would have studied the structure and functioning of human cell, right? So how it works and what are the different components inside the cell, all those things you would have studied. So this electrode provides the chemical structure for cellular reaction. So electrolytes at the cell membrane allow the transmission of electrochemical impulses in the nerve and the muscle cells. So this is what we have seen in this video. So your nervous system will help you conduct the electrical signals from the brain to the parts of the body. So in order for the neurons to transmit the signal inside the cell, this electrochemical components will help the transmission of this electrical impulse. Okay. So these electrolytes act as a catalyst for many enzyme reactions that are necessary for cellular metabolism. So whatever the metabolism happening inside the body, these electrolytes are the main components. So what is a biopotential? Ionic voltage is produced as a result of the electrochemical activity of the excitable cells. So whatever the neuronal action which is happening inside the body. So the body is made of lot of neurons, right? So whatever the neurons are transmitting the signals, that will cause the biopotential. So how we are going to measure the biopotential? Using transducers or sensors to convert the ionic potentials to electrical potentials. So basically the electrolyte will have the chemical components. That chemical components and its reaction you are going to convert into electrical potentials. So that electrical potential we are going to measure. For example, electrocardiogram, electromyogram, electroencephalogram, electroneurogram, electrooclogram, and electroretinogram. So what is a gram? Does anyone remember the difference between gram, graphy, and graph? The first class itself, I think I have discussed about the difference, right? Does anyone remember what is electrocardiogram? Are you guys asleep?
Ma'am,
Yeah,
recorded output signal, ma'am.
very good, very good. So gram is the recorded output signal. Graphy is the technique, and graph is the instrument. Okay, so electrocardiogram is the recorded output. Okay, so all these biopotentials are measured using the sensors. Okay, so this is the cell. So a typical cell. So if you look at the typical cell, you will have more electropositive charge outside and electronegative charge inside. So if you are going to measure the potential, which if you are going to check the potential of a cell at its resting state when it is not conducting any signal, for example, if I am just lying in the bed and I am not doing any movement, in that case all the cells of my hand and legs are in resting potential stage. If I am going to take a walk or if I am going to run, in that case my hand and leg muscles are moving. So for the muscles to move, you need some electrical impulse from the brain, right? So in that case the cell will be in a electropositive charge. Okay, so in that case there will be an action potential. So this resting state and its potential is called a resting potential. So okay, so this is the depolarization process. So before I explain this one, let's just continue with the video. So up to this part we have seen. So the resting potential is whenever the body is in resting state, when the neurons are not transmitting any signal, it's called as a resting potential.
of approximately minus 70 millivolts.
So the resting potential is approximately minus 70 millivolts for a normal
This
cell.
means that the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to establishing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane and the bulk solution on either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electron gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in the membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require a signal to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through.
So this part is very important. So when we have a cellular structure, the cell has a membrane, right? So the outer layer of the cell membrane has different channels. So they have mentioned three different channels: voltage gated channels, ligand gated channels, and mechanically gated channels. So voltage gated channels will allow the ions to pass through whenever there is a voltage change. So which means whenever there is an action potential transmission, in that case your voltage gated channel will open and close. So basically this cell membrane has different types of walls. So let's say you have a motor and a wall, motor connected to a wall. In that case, to fill the tank, only when you open the valve the water will run through the pipe and then reach the tank, right? So if you are closing the valve, the water won't reach the tank. So similarly, this walls like structures present on the cell membrane is called a channel. Okay, so the channels are the structures which act as a wall to prevent the movement of the ions inside and outside the membrane. So second one is ligand gated channel. So the ligand gated channel will open and close whenever there is a ligand attached to the channel. Okay, so only if a particular ligand is attached to that, it will open, or else it won't get open. It will be in a closed state. Some channels are always open. Okay, and last one is mechanically gated channel. So all these channels play a vital role in the transmission of electrical impulses through the neurons. Okay,
Small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion, i.e. voltage-gated sodium channels as well as voltage-gated potassium channels. As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a graded potential. Graded potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open and a graded potential occurs, the neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pump, which uses the energy generated by ATP hydrolysis to actively transport ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell, where its concentration is higher, and potassium is transported back into the cell, where its concentration is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentrations we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining these ionic balance in neurons is so important that this process can account for 20 to 40% of the brain's total energy use.
So now this is a very important point. So we saw different channels, right? So for potassium we have a separate channel, sodium we have a separate channel, but this sodium potassium pump mainly acts as a key uh, key factor for maintaining the homeostasis, that is the equilibrium of the cell. So you can see that it uses around 20 to 40% of the brain's total energy use, which means that whenever there is a potential change happening, the this sodium potassium pump, what it will do is it will send two potassium inside and 3 sodium ions outside to maintain the electronegative state. So to maintain the cell at its resting potential, this sodium potassium pump will act as a main factor, and this process accounts for 20 to 40% of the brain's total energy use. So this sodium potassium pump is triggered by the process of ATP hydrolysis. So hydrolysis is the process where the ATP, adenosine triphosphate, is broken down to ADP plus a phosphate ion. So whenever it is broken down to ADP plus a phosphate ion, this sodium potassium pump will get activated. Okay, so this sodium potassium pump is very important thing. And okay, let's continue with the video.
Only when the resting membrane potential and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from minus 70 millivolts to the threshold voltage of minus 55 millivolts or higher, this triggers an action potential at the axon hillock, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position, and sodium rushes into the cell because of the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive as it approaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0, up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gate shuts, making the channel inactivated. This stops the flow of sodium ions into the cell. The change in membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place. And the pump reestablishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential.
So I think now the process is more clear. So whenever you're asked about the action potential graph, you have to draw this graph which is given at the top. So usually the cell is at a resting potential of minus 70 volts. Whenever there is a potential which crosses the threshold, so the threshold potential is minus 55 millivolts. So whenever it crosses the threshold, then this neurons will transmit the action potential. So what happens is there is a depolarization happening. Depolarization means the cell is becoming more positive, electropositive. So in that case, just give me a minute. Yeah, sorry. Yeah. So in that case, your cell is becoming more electropositive. So you can see that the potential is reaching from a negative to zero, and then it crosses zero and reaches a positive electropositive state. So when it reaches the electropositive state, the cell is having more positive charges inside and negative charges outside. So again, it has to restore the resting potential. So this, from the peak when it reaches to the negative, this region is called as repolarization. Again, it is going back to its resting potential stage. Okay. So you have to know between depolarization and repolarization. These two terms are very, very important. Depolarization is it goes from resting potential to action potential. Repolarization is it goes from action potential to resting potential. So when this repolarization is happening, when it is going down to the electronegative state, what happens is this sodium potassium pump, which is our main key factor, this pump will throw the excess sodium ions outside and bring back the potassium ions inside, so that the electronegative state is maintained. Okay. So before it reaches the resting potential, the cell will be in electronegative state for a short period of time, and then it will go back to its resting potential. So at this point, your sodium potassium pump is your main factor. Okay. So whenever you are asked about this action potential graph, you have to draw this graph and then you have to explain. Okay. So first sodium channel will open, next potassium channel will open, and then sodium potassium pump will restore the equilibrium. Okay. So this is the graph.
Action potential for a particular neuron. That is, the maximum voltage in one neuron during an action potential never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen.
This is very, very important. For each and every neuron, this action potential will be same, but it never exceeds the threshold value. For example, if it reaches plus 30 millivolts, all the neurons will have plus 30 millivolts. So this rule is called all or nothing rule. So basically how it works is, for example, if I am going for a jogging, in that case my hand and leg muscles will be activated, so all the neurons will be having some action potential transmission. So I am going for jogging. But if I am running and I am running faster, in that case this potential will not exceed this maximum value. All the neurons will have the same action potential, but then more neurons will be activated. So that's how this neuronal signal transmission works. Okay. So when I'm jogging, only a few neurons will be activated, so only a few muscles will be activated in my hands and legs, so I'm running slowly and I'm jogging. But if I'm going to run for a longer distance, or if I'm going for a high speed running, in that case more neurons will be activated, but then all the neurons will be having the same amount of action potential. Okay. It's more like your electrical circuit. So for example, if you are having a tube light in your house and if you are connecting it to a circuit, so the current flow will be the same, right? So for example, if it is 230 volts, in that case the circuit will have only 230 volts, it won't exceed that, right? So similarly in our body's nervous system, this biopotential will have only that maximum value, it never exceeds the maximum value. Okay. So this one you have to remember very clearly. Are you able to follow what I'm saying, guys?
Ma'am.
Yes. Okay, good.
What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain, and less frequently in response to a gentle breeze. Some axons transmit action potentials faster than others. One variable that increases conduction velocity is the presence of myelin sheaths around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signal appears to jump along the part of the axon covered by the sheath. In the peripheral nervous system, the sheaths are formed from glial cells known as Schwann cells. There are small gaps between Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous system, the sheaths are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a graded potential, and a stimulus above the threshold creates an action potential and the neuron fires.
Okay, so that's about the action potential. Let me just go back to the slide. Okay, so now we will understand this picture more clearly. Okay, so previously we had negative charges inside the cell, and then when you measure that, it's called a resting potential. And now you're having movement of ions across the cell membrane, right? So your sodium is entering inside and potassium is going outside. So sodium has a higher positive charge. So when it enters the cell, you will have a positive charge inside the cell, which causes the action potential. So when you measure that, so the cell will have more positive sodium ions inside and the potassium ions are outside. So this will cause a positive action potential. So the range is around 20 millivolts to 30 millivolts. So this is the depolarization. Okay, so this process of having positive ions inside the cell is called as depolarization. So you should know the difference between depolarization and repolarization. Again and again in 2 mark question you will get this question. Most of the time students will write it wrongly. So depolarization is having electropositivity inside the cell. Repolarization is coming back from action potential to resting potential. Okay, so this is the typical waveform of your action potential which we just saw, right? So in the video they explained it very clearly. So you have a resting potential which is minus 70 millivolts, and then the depolarization happens. So more sodium ions are moving inside and there is an action potential which is plus 20 millivolts, and then again the cell comes back to its equilibrium stage. So here you have a repolarization happening, and then here it doesn't just go back to its resting potential, there is a hyperpotential stage. So at this stage this sodium potassium pump will act as a key factor to restore the normal resting potential. So and then the cell will come back to its resting potential stage. Okay, so this is the action potential curve. Is everyone clear with this curve? Yes.
Yes, ma'am.
Okay, very good. So this is a very important question. Okay, so either you will get about question about action potential, or will be asked about depolarization and repolarization, or you will be asked a question about resting potential and membrane potential. Okay, so membrane potential is the difference at the cell membrane. Okay, so this is the same graph with respect to the muscle contraction. So I said about jogging and running, right? So whatever the process which is happening inside the body, whatever the signal, whatever the cell metabolism, everything is connected to your action potential, right? So whenever you have a chemical gradient, you will have some electrical gradient happening in the body. So that's why it's called as electrochemical gradient, which we saw in the video. So this graph connects the muscle contraction with your action potential. So when you study about electromyography, you will study about this in detail, which you'll be studying in your second unit or third unit, I think. Yes, you will be studying in your second unit. So anyway, I'll explain this graph. So here you have this action potential curve, right? So during the action potential, what happens is your nervous system is always connected to your muscular system. So not just muscular system, all the cells and organs in the body is connected to your nervous system. So at the junction of your nerves and muscle, there is something called as neuromuscular junction. You would have studied in your anatomy and physiology. So whenever there is an action potential happening, the muscles will start to contract and the muscle contraction occurs. So if you see here, at the top of the action potential peak, your muscle contraction is starting. So this curved line, if you can see here, this dotted line, so the muscle contraction starts at the peak of the action potential and the muscle is at a contraction stage. The muscle reaches its peak contraction and again it comes back to its relaxation state. Okay, so whenever there is a contraction, there will be also an relaxation. So when you are flexing and extending your hands or legs, there will be both contraction and relaxation happening. So that's called a sliding theory of muscle contraction, which we'll see in a EMG. Okay, so EMG topic when we are discussing, we'll study all about that. So this is how your muscle contraction is related to your action potential. Okay, and again this is a small picture explaining resting potential and membrane potential. So this you would have understood more clearly from the video which I had shown. And again the same action potential pictures. So this is just to recap. So here you have a sodium channel. So sodium channel opens and the sodium begins to enter, and then the potassium channels open and the potassium is leaving the cell. So more sodium enters and the action potential is reached, and then potassium is keep on leaving the cell. So the cell becomes more electropositive, and then again when it comes back to equilibrium, the sodium and potassium channel close, and then the sodium potassium pump will uh, throw the extra sodium outside and bring the potassium inside. So again the cell goes back to its resting potential stage. Okay, so that's about your action potential, resting potential and membrane potential. Okay, is that clear? Yes, no?
Yes, ma'am.
Okay, great. So next thing is types of electrodes. So types of electrodes is your second half of the topic in your second section. Shall we proceed with types of sensors today or we'll discuss it in the next class? I'm asking you guys.
Next class, ma'am.
Class proceed பண்ணலாமா? Okay. Okay then. Then I'll give you a small activity now. Okay. So what about this chart activity I said? Have you guys finished it? Yes, no? You were supposed to finish it by Sunday, right?